Hey everybody, this is going to be our talk on membranes. This is one of our last few lectures. This is our last little bit of new content. So let's go ahead and get started. So membranes are structures that have many cell functions. For example, they are barriers to toxic molecules, and you can also think of them as a barrier to non-toxic molecules. They help accumulate nutrients, so they basically help what you want to keep in, or help stuff stay in and help stuff stay out. That's a nice way to think about them. They carry out energy transduction, facilitate cellular motion, assist in reproduction, modulate cell transduction, and mediate cell-to-cell interactions. So you can kind of think about them as they are great communicators. They are great at sensing everything outside and kind of communicating that inward, but then also communicating what is inside out. Now, when it comes to membranes, there's three major structures that you need to be familiar with. They are a micelle, a bilayer, and a liposome. If we look at a micelle, what we see are a bunch of, well, individual units, and they are wedge-shaped. Now, these individual units have a polar head group and a non-polar tail. The same is true of these other guys right here. Now, it is significant to note the differences between them. The difference between one of the building blocks for a micelle and a bilayer. Well, this is one non-polar tail, whereas your bilayers, they're typically going to have two non-polar tails. Now, these tails are, let me go ahead and draw a example of one. And let me get my eraser tool back out. Nope, not my skip ahead tool, my eraser tool. This is going to be one of the tails of these. So this is a hydrocarbon, a non-polar hydrocarbon. Loves not being near water. Now, with that in mind, when you look at a micelle, the interior of a micelle, well, that's very non-polar. It's got a bunch of, or it's very hydrophobic. It's very non-polar. If you look at the interior of a micelle right here, well, that's also going to be non-polar. If you look at this last of our third major structures, this is a liposome. A liposome has, well, basically a bilayer, and then on the interior of it, it has, which is highlighted right here, an aqueous cavity. So the kind of the core of a liposome is aqueous. So that is a polar region. Now, whenever we look at a bilayer, and if you look back at this, well, we've got micelles, bilayers, and liposomes. We're going to focus primarily on bilayers, in part because those are when you're talking about a cell, you have a cell bilayer, and a cell is large enough that the entire interior of it is aqueous, and we don't look at it on a small scale the way that we look at a liposome. So when we zoom in on a part of a lipid or of a membrane of a cell, we're looking at and we're focused on a region of a bilayer. A liposome, definitely they are involved in transport of proteins and positioning molecules in different places. But for our purposes, we're not going to be worried too much about liposomes. We're going to be focused on bilayers. Okay. Lipid mobility. So if you look at this, we've got one of those building blocks for our bilayer. And well, here's our bilayer. Here's our two different layers. If we think about this space that I'm kind of scribbling in right here, I'm going to call that extracellular. This extracellular space, and then this would be our cytosol down here. I want you to be familiar with and comfortable with this terminology. There are two sides to a bilayer. They are our outer and our inner leaflets, L-E-A-F-L-E-T. So the outer leaflet and the inner leaflet are the two sides of our bilayer. If you look at one of these components or one of these individual that could be a glycerophospholipid. One of these glycerophospholipids. It's got the polar head group and the non-polar tail. Well, this glycerophospholipid can go from the outer leaflet to the inner leaflet, and this is going to be very, very slow. This is going to be very slow whenever it is not in an enzyme catalyzed, or it is not catalyzed. This movement is not catalyzed by an enzyme. Now, one of the reasons that it's going to be slow is think about this polar head group. This polar head group right here has to end up on the opposite side. So think about the path that it's going to take. It's going to be moving from here all the way to here. So that's a polar group moving through a very non-polar or a very hydrophobic region. So the movement is, it's working against kind of the, I guess you could say, the preference. It's moving against, it's moving in the opposite direction of what is easiest to do. It's a difficult movement, moving from a kind of foreign region. Next up, we have another type of movement where we have this glycerophospholipid that I'm going to highlight right here, and it goes from one position to the next position. This polar group remains on the outer leaflet, and it's almost like, the way I always kind of think about this movement is lateral diffusion is, let's say you go to a concert and you want to go from the very back to the very front of the, the general admission standing area. You're going to face a little bit of resistance, but you're just one person moving within the crowd. I don't have really a good comparison for transverse diffusion, but that lateral movement, yeah, you're going to bump into people, but you're going to be able to get where you need to go. We're going from we're staying in that outer leaflet there. Now, this principle and this idea is really important, and it stems from what we talked about when we discuss individual fatty acids. So fatty acids, there are a couple of things that were important about them. And one thing that was important about them was that as the number of carbons in a hydrocarbon side chain increases, so too does the melting point. And what that means is that if you got a bunch of, let's say you got a bunch of C16:0. This is going to have a melting point, and I'm just making an estimation here. It's going to be about 56 degrees Celsius. Okay, so this is going to be a solid. That's going to be a solid at room temperature. As you get to 56 degrees Celsius, what's going to happen is that solid is going to liquefy, and it's going to form a liquid. Now, the same thing is true of membranes. Membranes are sensitive to changes of temperature. And at a higher temperature, they're going to be in this fluid state. So higher temp. And in this fluid state, I'll ask you to look at this structure right here that I put a box around. It looks pretty chaotic. And you're not wrong. It's absolutely chaotic because all of those individual glycerophospholipids that make up that membrane, they're mixed in with one another. At a lower temperature, by comparison, well, you're going to form a solid. So each of these individual components will find their place. They will stop. They will freeze up. So each of these individual glycerophospholipids, they will produce something that overall is very rigid. So when you add heat, that's add heat, not feet. So when you add heat to this polycrystalline solid, or this polycrystalline state of a lipid bilayer, it's going to transition to more of a liquid or a fluid state. And this is referred to as the transition temperature. So what I want to bring back is, for individual fatty acids, we have a melting point. For a membrane, we have a transition temperature. And below the transition temperature, the bilayer becomes a gel-like solid. So at a lower temperature, so below the transition temperature, that's what you get. above the transition temperature, the bilayer is known as a liquid crystal. So above that temperature, you're going to have, that's where you're going to have that more liquid form of it. Now, the transition temperature is dependent on chain length and the degree of saturation of the component fatty acids. Okay, now let's read that and think about that for a second. Transition temperature is dependent on chain length and degree of saturation. Well, if we think back to individual fatty acids, I gave you one. If you had a fatty acid that had a melting point of, it was 16 carbons long, had a melting point of 56 degrees Celsius, and then you also had C16:3. What's our melting point of this? Probably going to be like -8 degrees Celsius. So fatty acids are sensitive to temperature change just as entire membranes are. Now, cholesterol will broaden the temperature range of that phase transition. So cholesterol is a compound. You remember, cholesterol has 5 6-membered rings and 1 5-membered ring. 6 5-mem and 1 5-mem. 5 6-mem, sorry. What this is going to do is it's going to insert itself. So you're going to have a cholesterol molecule right here. And that is going to have an impact on the stability of a membrane and basically the overall structure of that membrane. So it's going to change the temperature range of that phase transition. Now, this right here is one of my favorite slides to discuss because I feel like it's a great application of that phenomenon. Now, there's a lot to unpack here, and I know that I say that a lot, but what I want you to focus on are these 2 fatty acids, myristic acid and palmitic acid. These are 14 and 16 carbons long, respectively, and they're completely saturated fatty acids. So I'm going to label these as saturated. Now, what this table or this figure is showing is that when E. coli was cultured at 10, 20, 30, and 40 degrees Celsius. Well, first of all, E. coli can grow at those different temperatures. E. coli has an optimal doubling temperature of approximately 37 degrees Celsius. It can grow at 10, 20, and 30 degrees Celsius, so below that ideal temperature, but it's going to grow a little bit slower. Now, with that in mind, when we grow E. coli at 10 degrees Celsius, what we're going to see is approximately, and I'm going to add these two together, 22% of the total fatty acids were saturated fatty acids. Okay, that's good to know. That's just like, let's mark that off. Okay, so 22%. Now we don't know what to compare that to, or we don't have any kind of basis for comparison. However, what we can do is we can go over here and say, all right, well, when we culture E. coli at 40 degrees Celsius, what happens? Well, at 40 degrees Celsius, we have approximately 56% of the total fatty acids are saturated fatty acids. So at 40 degrees Celsius, we have 56% of our fatty acids are, well, are saturated. Now let's think about this. Saturated fatty acids at lower temps. Well, at a lower temperature, we're going to say they are solid, or they are more solid. Because, you got to think about, it's an equilibrium. And whatever I say in equilibrium is, at 32 degrees Celsius or 32 degrees Fahrenheit, is all water a solid? No, it's not. Strange. Oh, sorry about that, everyone. Okay, so then what about saturated fatty acids at higher temps? Well, it's more likely going to be a liquid. Okay. So, it's kind of like there's going to be some sort of sweet spot where there's a mixture of solids and liquids. And then if you crank the temperature way up, you're going to have a higher percentage of those fats that are a liquid. And then at a lower temperature, if you crank it way down, the higher percentage that are solids. Now, with this in mind, however, what I really like to see is or we can talk about trends. Increase temp, and I'm going to bracket sat. That's my way of saying saturated fat concentration goes up. Okay. So then you could also say it as decrease the temperature and your saturated fat percentage goes down. Now let's look at palmitoleic acid and oleic acid, 16:1 and 18:1. These two are unsaturated fatty acids. And we know that unsaturated fatty acids at lower temps, well, yeah, they're going to be, or actually, let me say, unsaturated fatty acids have lower melting points. Unsaturated fatty acids at lower temperatures, you're going to see a higher percentage that are liquid than saturated fatty acids. So now let's look at, I'm going to change colors on this actually, just to give a good contrast. So I'm going to bracket this. (26 and 38). That would be 58, 64% of fatty acids are mono unsaturated fatty acids at 10 degrees Celsius. So we've got like almost a 3-to-1 ratio of unsaturated fatty acids to saturated fatty acids at 10 degrees Celsius. At 40 degrees Celsius, only about 21% of fatty acids are unsaturated. So we have this inversion. At lower temperatures, lower temp equals higher percent unsaturated fatty acids. Do you still have some saturated fatty acids? Yes, you do. At higher temps, do you still have some unsaturated fatty acids? Yes, you do. But you see a shift from a high percentage of unsaturated fatty acids and a low percentage of saturated fatty acids to a high percentage of saturated fatty acids and a low percentage of unsaturated fatty acids. So there's this kind of trade-off almost of, well, what do we want to have? And the cool thing about this is that it's almost like you hit a sweet spot of sorts. I always think about this as, let's see, you have your saturated and unsaturated fatty acids. Those two combine to give us something, to give us a membrane. What's that membrane going to do? That membrane has to do all of these things. It has to act as a barrier. It has to modulate cell transduction, assist in reproduction, facilitate cell motion. So it has to do all of those things. So I always use the term structural integrity. That membrane has to maintain structural integrity. So it's not like at 10 degrees Celsius, it's super rigid, and at 40 degrees Celsius, it's not super rigid. Instead, the tension and the rigidity of that membrane, whether you're at 10 degrees or 40 degrees, is effectively going to be the same. So how do you do that? Well, you do that by having a higher percentage of unsaturated fatty acids at low temperatures, or when you're at a low temperature. Whereas at a high temperature, you don't need so many of those unsaturated fatty acids. Instead, you need more saturated fatty acids. So I always kind of think about this as these two types of fatty acids are serving different purposes. The question is, what is the purpose? Like, what are we trying to build? We're trying to build a membrane with a certain amount of strength. If we're at a low temperature, we want it to be just as strong as if we're at a high temperature. And the cell responds by modulating the percentage of these different fatty acids because they have different physical properties. Now, this brings up the terminology that I was talking about a little while ago, inner leaflet and outer leaflet. So if we think about a lipid bilayer, the outer leaflet is the portion that's exposed to the extracellular space. And the inner leaflet is exposed to the inner portion of the cell. Now, what we have a couple of different major molecules. Sphingomyelin, phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine. These are all glycerophospholipids. Sphingomyelin is a sphingolipid. What we've got is these two, sphingomyelin and phosphatidylcholine, that are found on both sides of the membrane, but in particular, there's a greater percentage of them found on the extracellular or on the outer leaflet. So they are kind of used for communication. Whereas phosphatidylethanolamine and phosphatidylserine, those are more likely going to be found on the inner leaflet. Now, the thing is, when you look at a slide like this, think about this as basically where different molecules are found or what they're exposed to or when they're exposed under specific conditions. Because, let's go back a second. Are lipids mobile? Yes, they are. If something is found on the outer leaflet, can it be moved to the inner leaflet? Yep. Can something be moved from the inner leaflet to the outer leaflet? Can. So under, stress conditions, these numbers might flip-flop. One takeaway, however, that's important is total phospholipids are going to be found on the inner and outer leaflet equally. Now, that lipid asymmetry, phospholipids are synthesized on or near the interior of the membrane. There are two mechanisms to get those phospholipids from one side of the membrane to the other. Flipases, I kid you not, that is one of the most fun enzyme names ever. Flipases catalyze the flip-flops of specific phospholipids in what is known as facilitated diffusion. So here what we have is this uncatalyzed movement that we talked about previously. This is slow. In fact, it's so slow that it's written in black here and written very slow. There is an enzyme, or there are enzymes known as flipases that move things from one side of a membrane, the outer to the inner leaflet or the inner to the outer leaflet, and that's going to happen very quickly. Your lateral diffusion, that's going to be even faster. Now, flipases tend to equilibrate the distribution of their corresponding phospholipids across a bilayer. But like I said, under stressed or threatened conditions, it might be a 3-to-1 ratio. It might be, as we see here, all of your phosphatidylserine is on the inner leaflet. Well, under certain conditions, that might all be transitioned to the outer leaflet. Phospholipid translocases transport specific phospholipids across a bilayer in a process that is driven by the hydrolysis of ATP. Translocases can establish a non-equilibrium distribution of the phospholipid. Now, membrane composition. Biological membranes contain proteins and lipids. Those are not all, but those are your major components. The proteins that catalyze a reaction, mediate the flux of nutrient and wastes, and participate in relaying external information to the cell. Your protein to lipid ratios vary with your membrane's function. Here we've got a table where we look at the composition of different types of membranes. We look at E. coli. Well, E. coli doesn't have any sterols because they can't make cholesterol. Liver cells, we've got 45%, 25, 27%, and 25% of your proteins, phospholipids, and sterols. The most important takeaway from this is, well, your protein to lipid ratios vary with that membrane function. So the membrane structure and assembly is pretty darn important to understand. So the phospholipid bilayer is considered a fluid matrix. And there we go. The bilayer is a two-dimensional solvent. And this picture right here captures the major parts of your lipid bilayer. So let's go ahead and fill in some blanks. We've got our outer leaflet and our inner leaflet, corresponding to the outside and the inside of the cell. Lipids and proteins can undergo rotational and lateral movement. So they can go, or they can go whoop, boop, from one side to the other. Membranes are asymmetric, but this captures your major components and where they're going to be found. So typically, your oligosaccharide chains of a glycoprotein, they're going to be found on the outside of the cell in the outer leaflet. Glycolipids, an example of this would be something like a ceramide. This is a sugar plus a fat. And if you think about, sorry, not ceramide, a ganglioside. If you think about a ganglioside, what that consists of is you've got 1, 2, 3, 4, 5 sugars, one of those being sialic acid. And that's linked to a hydrocarbon chain that is embedded into the membrane. So a ganglioside is typically going to be found in the outer leaflet. Then these little hot dog type structures right here, those are your sterols, like cholesterol. And those can be found on one side or the other side of the membrane. So they can be found on the inner side or the outer side of the membrane. Now, with that in mind, let's talk about some of the other major players. You have membrane proteins. And these membrane proteins can span the entire membrane, or they can be found on just one side. So here what we have are two proteins. These are integral membrane proteins. These are proteins that are embedded within the membrane, and they have a part of them that is on the outside of the cell, and then a part of them that is on the inside of the cell. These, in particular, have alpha helices that are embedded within the membrane. And those are what you're going to find. Now, in addition to those integral membrane proteins that kind of span a membrane, if you look, there's a couple of other proteins. These kind of they look like balloon animals, really, to me. These balloon animal proteins, what you'll notice about them is that this one right here and this one right here, or what I want you to notice about them is that they're kind of unique in that they're not embedded in a membrane and doesn't, however, that they do have kind of linkages or connections that basically make them a part of membrane, even if it is kind of tangential. It's like, yeah, they're a part of the membrane, but they're not inserted into the membrane. So when we look at these other proteins, the non-integral proteins, we've got these peripheral proteins. That a peripheral protein can be, well, the peripheral proteins do not have any of their amino acid sequence in the membrane. So they are associated with the membrane, however, in a different manner. A peripheral protein can be covalently linked to a lipid. In addition to that, a peripheral protein can have some sort of modification that links it to a polar head group. Now, this is another kind of cartoonish model of a protein, of a membrane, and maybe the scale is definitely a little bit different, but it's effectively saying the same thing and presenting the same information, where things are and how they are embedded within or associated with a membrane. Now, integral proteins are associated very tightly, and in order to kind of free them or liberate them from that membrane, you're going to have to use something like a detergent. And I was about to underline that, but I realized blue ink is what I have right now, so I'm going to switch to white ink. You're going to have to use a detergent or a chaotropic agent in order to separate these from the membrane. Now, with that in mind, an important thing to know about a detergent is that detergents have a couple of different properties, and they're classified as being either ionic or non-ionic detergents. And basically what they're really good at is kind of breaking up by using hydrophobic interactions. Chaotropic agents are going to disrupt hydrogen bond networks. So these two things are either it's a detergent or a chaotropic agent. Effectively, what they're going to do is they're going to undermine the integrity of the whole system. Like, oh, hydrogen bonds held it together? Not anymore. Detergents held the whole system together? Not anymore. So those are the big deals with integral proteins and how they're associated with. Lipid linked, however, tend to be globular. And we're going to look at a couple of examples of those in just a moment. And peripheral proteins are also globular-like and are easily dissociated. So here what we have is a couple of different ways that proteins can be associated with a membrane. And what I'd really like you to focus on are these two down here. Type 5 and type 6 transmembrane helical proteins. Type 5, what I want you to notice about that is it doesn't have that cartoon image of an alpha helix, but instead it has a modification, or it has a lipid that is inserted into the membrane, and then that protein is kind of hanging off of it. Type 6, on the other hand, well, that's got an alpha helix that's embedded within a membrane, and then it's also got this guy right here. Those kind of look like, I don't know, like little cartoon spider web legs or some spider legs or something like that. Maybe they don't, I don't know. No, they look like the design on Charlie Brown's shirt. But those are lipids. So that is a modification to this protein that has hydrocarbon side chains. Those hydrocarbon side chains are inserted into the membrane, and that whole thing is anchored there. So type 5 and type 6, the difference between those two is basically type 6, they have an alpha helix that is spanning a membrane, whereas type 5 doesn't have an alpha helix. Type 6 has the alpha helix and it has that lipid linkage. Our other types are really type 1 and type 2 transmembrane proteins. The only difference between them is the positioning of the N-termini. Type 1 has the N-termini on the outside of the cell, whereas type 2 has the N-termini on the inside of the cell. Type 3 and type 4, the difference between these is type 3 is one singular polypeptide with multiple transmembrane, alpha helices that span this membrane, whereas type 6, that's multiple polypeptides that aggregate together. They're all helices, but they aggregate together to form this complex. So the difference between 3 and 4 is 1, I'm going to write PP for polypeptide, or more. So that's the only difference there. Now, or that's the fundamental difference between them. There are also proteins that span a membrane. There are transmembrane sheet proteins. And these transmembrane sheet proteins, well, these are beta sheets that form, that kind of wrap around and basically make, it's almost like a pore or a cavern that spans a membrane. So this is one of the things about these membrane proteins that make them complicated. If we look at alpha hemolysin toxin, we've got this what's called a beta barrel. So that's a beta sheet that makes a cylinder structure. Then let's look at the top-down view of it right here. That's effectively what we've got. We've got a hole within this transmembrane protein. That's what we're effectively doing here. Now, what these are able to do is these can make channels, and these can open and close. They can more or less be open so that nutrients can flow through, but then they can close off to basically stop the flow of things from going through that membrane. Now, lipid-linked proteins, these structures right here and here, the Charlie Brown shirt design, that's what these guys are. These are carbon structures that are 15 and 20 carbons long. Farnesyl and a geranyl or geranyl. These are both lipids that can be embedded into a membrane to anchor a protein. Some proteins are associated with membranes through covalent attachments to lipids that anchor that protein. Now, there are 4 major types of other linkages. Thioester-linked fatty acyl anchors. These are always going to be palmitic acid, and a palmitoyl group can be removed by an enzyme known as palmitoyl transferase or thioesterase. Excuse me. This you're going to take basically like I think palmitic acid is 16 carbons long, roughly like a 16 carbon structure, and add it to a cysteine or a serine. I want you to know these. The specific amino acids. Now, the cool thing about this is that this can be cut. And whenever that is cut, or I believe it's a cut there, whenever that group is cut off, well, then this protein can float on and do whatever it needs to do. It's embedded into a membrane, but then whenever it's needed to not be embedded in a membrane, you cut, and then it's released. Now, another example is this guy right here. Glycine. So a glycine can be modified, a myristic acid or a myristoyl group can be added, and that same thing can be broken and the protein floats off. And it's floating off within the cell and can do whatever it needs to do. Farnesyl groups like this guy right here is also a way that a protein can be anchored to a membrane. And whenever the time is right, that anchor can be cut off. And before I proceed, I really want to bring up, if I remember, I'm like 98% sure on this. If you've ever heard of the aging disorder known as progeria, progeria is related to a protein receiving a fatty acid modification. So it's associated with a membrane. And then during certain phases of the cell cycle, there's an enzyme that's supposed to come along and cut that to release that protein or to release a protein. But progeria is highlighted by the enzyme doesn't do that. And so what happens is that protein that's supposed to be released never gets released. And that leads to cell cycle checkpoints that are missed. So normal replication and normal replication of cells doesn't take place as it should. And what ends up happening is that with this disorder, progeria, the way that it works is that people afflicted with progeria age like 4 times as fast as they otherwise would. And so this disease is caused by a modification that doesn't get removed. So this protein is anchored in the membrane when it's supposed to be released to go to the nucleus to turn on gene expression or turn off gene expression. There are also GPI-linked proteins, and here's this anchor right here. This molecule is really complicated. So this right here is our polypeptide. These are our polypeptides. These are our proteins. And within those proteins, they are specific amino acids. Cysteine or a serine gets a palmitate. Glycine gets a myristic acid. And cysteine gets a geranyl residue. And those get associated with a membrane. Okay? So this balloon animal, this red balloon animal type of structure, that's your protein. Here's a protein. That protein gets a phosphate group, which then gets a network of sugars added to it. That network of sugars gets and inositol. If you remember inositol, that's one of the head groups that can be added to a glycerophospholipid. That inositol has a phosphate. That phosphate, oh, whoa, lo and behold, that is a glycerophospholipid. So we have a glycerophosphoinositol linked protein. This is on the outside of the cell, and this is projected outwards. So this is a way in which a protein is displayed on the outer surface of the cell. This protein can then be used for cellular communication because it's on the outside. This whole structure that I've shown basically from here to here is this thing, but we're seeing it in a different way. So here's our protein. Here's our phosphate. Here's our sugars. Here's our inositol. Here's our phosphate. Here's our glycerol backbone. Here's our two fatty acids that are embedded in the membrane. Okay, so that's really going to wrap up everything that I wanted to get through with respect to membrane-associated proteins. So I hope this was helpful. And these last two slides, we don't have to. Oh, I did want to mention membrane rafts, and this is where proteins get concentrated. And whenever they get modified, they'll concentrate and form what are known as membrane rafts. Membrane rafts are rich in sphingolipids and cholesterol, but not phospholipids. A number of transport proteins are associated with lipid rafts. But that's going to do it. And I hope you have a good one.